Hello, welcome to biology lesson 5.05 over mitosis. Now, we have said in the past videos that mitosis is the process of creating, of a cell dividing and creating two exact daughter cell copies. And we're going to hone that definition a little bit. Actually, mitosis is the process of dividing the genetic information. Turns out that it's actually at cytokinesis where we create the two cells. But we want to talk about the four phases. We say we've got PMAT, P-M-A-T, prophase, metaphase, anaphase, and telophase or telophase. So when we start out at the end of interphase, so at the end of interphase, our cell, if we looked at it under the microscope, would look like this. And we would see a nucleus. We might see a dark area inside the nucleus called the nucleolus. We would have the cell membrane. When we get to metaphase, so one of the things I like to do is I like to draw a sketch for metaphase and a sketch for anaphase, and then let's look and see what happens. Can we deduce from those two what happens during prophase and telophase? If I go to metaphase, and if I go to anaphase, we're going to look at slides, and you're going to see images, and you're going to have to tell me if something's in interphase, prophase, metaphase, anaphase, or telophase. So that's what metaphase looks like, and the X's are chromosomes. And that's what anaphase looks like. So let me do a little key up here. These are chromatids. So what do you see with metaphase? Well, the chromatids, or we could say the chromosomes are lined up in the middle. Chromosomes are lined up in the middle, and we have M for metaphase and M for middle. So that's easy to remember. In anaphase, the chromosomes are being pulled away from each other towards opposite ends of the cell. So chromosomes, I don't know why I have trouble spelling chromosomes, pulled away from each other. A for away, A for anaphase. So these are super easy to identify. M for middle, chromosomes in the middle. So if you look at this and you look at this, what has happened? And actually, prophase is such a busy phase that they sometimes divide it into early prophase, middle prophase, and late prophase. We're just going to do prophase, and we're just going to talk about it overall. So take a second, pause the video. What's the difference between this cell and this cell? Well, okay, here we go. So hopefully you saw that this cell has a nucleus. You can't see, there's no trace of a nucleus over here. And remember, the nucleus is surrounded by something called, I don't know if we've talked about this before, but the nucleus is surrounded by organelle. A nuclear membrane. So the actual organelle there is the nuclear membrane. And from here to here, that's gone. So the first thing one of the things that happens in prophase is the nuclear membrane dissolves. Nuclear membrane dissolves. Also, do these chromosomes show up over here? No. So somewhere between here and here, these chromosomes appeared as if by magic. Well, they were always there, but here it's in chromatin and over here it's in chromosomes. So the chromosomes have to wind up. From chromatin. Well, what's the other thing? What's all this stuff? These are called centrioles. And these are called spindle fibers. I'm not going to test you over that, but these are considered an organelle. Centrioles are considered an organelle, and they build these spindle fibers that attach to the chromosomes. So this whole shebang here and here had to set up. It's not apparent there. So the centrioles and spindle fibers form and line up. So those are all things that you don't see here at the end of interphase, and you do see here. So something you might notice if you were looking at a cell in prophase, it might look like this. You might see a cell, and you might just see like a clump of chromosomes kind of hanging out. So the nuclear membrane's gone, but the chromosomes have wound up, but they're not lined up yet. And then you might see, if it was a little bit later in prophase, or you might see this. Where the spindle fibers and centrioles have started to form, but they're not in the middle yet. All right, well, here's anaphase. So what we're doing is we're pulling apart these chromosomes. And at the end of this, my cell's going to look just like this. So how does this look different from this? Well, all the same things. So basically, all of these things have to undo. We have to unwind the chromosomes, we have to reform the nuclear membrane, and we have to take apart the centrioles and the spindle fibers. So we're going to, let's do this in purple. For telophase, we have to reverse this. So we have to reform the nuclear membrane. We have to unwind the chromosomes. And we have to dismantle or take apart the centrioles and the spindle fibers. It's really difficult to tell where if a cell is in telophase or cytokinesis. So if this is an animal cell, what we'll see is, I'm going to have to kind of just, you know what, let me white this line out. So I have a cell, and it's still considered, we haven't divided yet, and I would see a nucleus. So this is a nucleus, and this is a nucleus. If it's an animal cell, and these are animal cells, there is a protein that comes around the center, and it closes, it like wraps around, and this gets tighter and tighter and tighter. And it actually it'll form what's called a cleavage furrow, and it'll pull this cell membrane in and in and in until you end up with now our two daughter cells, which look, I mean, I drew them a little smaller because I was out of space. If you have a plant cell, this happens a little bit differently. So this pinches off. So let's say that these two cells pinch apart. And as they do so in cytokinesis, we divide the organelles and the cytoplasm. If it's a plant cell, let me see if I can squeeze this in. These are my two new nuclei. The cell will come in and it will build a cell wall. It'll form a cell plate. And it will actually build a cell wall between. So in this case, these get pinched, and as that's happening, we get a division. And in here, we build a cell plate between the two plant cells. We would end up then with two exact copies of a plant cell. All right, that is a summary. So our takeaways here are these are the stages of mitosis. And in mitosis, we're actually just dividing the genetic material. You'll notice we haven't created the two daughter cells until cytokinesis. Here I still have a single cell at the end of telophase. But what happens, we have to dissolve that nuclear membrane, form the chromosomes from the chromatin, set up the centrioles and the spindle fibers. So we have early prophase and then later in prophase. At metaphase, the chromosomes are going to line up at the middle. At anaphase, they pull away from each other. Telophase, we basically redo everything we had in prophase, and we end up with a cell that looks a lot like this. And in cytokinesis, we begin to pinch these two cells apart for animal or build a cell plate for plants. Let's write that down, that's plants. And at the end of the mitotic process, we have two identical ones. But notice during these, we're dividing the genetic info. Okay, that's all we're going to do for mitosis. The next video will be meiosis. Thanks for watching. Have a good day.